The biological activity of botulinum toxin type A is closely connected to its multi-domain protein structure. The active neurotoxin is organized as a heavy chain and a light chain connected by a disulfide bond. Although these regions remain associated during the early stages of cell entry, each performs a distinct function. The carboxyl terminal portion of the heavy chain forms the receptor binding domain. This region helps botulinum toxin type A recognize neuronal membranes through interactions with gangliosides and the protein receptor SV2. The amino terminal portion of the heavy chain forms the translocation domain. After the toxin enters an acidified vesicle, this domain undergoes structural rearrangement and helps deliver the light chain across the vesicle membrane. The light chain is the catalytic component. It functions as a zinc-dependent endopeptidase and selectively cleaves SNAP25, an essential protein in the neuronal snare complex. These three functional regions work in sequence. The binding domain determines neuronal recognition. The translocation domain supports delivery into the cytosol. The catalytic domain produces the intracellular biological effect. Structural integrity is therefore essential for complete activity. A protein may retain catalytic activity in a simplified biochemical assay, but show reduced cell-based activity if its receptor binding or translocation domain is damaged. Structural studies also support the design of recombinant fragments, inactive derivatives, binding domain constructs, and modified proteins for research. Depending on the scientific objective, investigators may study the intact protein or isolate individual domains to examine a particular molecular function. Understanding the relationship between structure and activity helps researchers select suitable analytical methods and interpret differences between biochemical and cellular results. Botulinum toxin type A is therefore best understood not as a single-function enzyme, but as a coordinated molecular system in which binding, translocation, and proteolysis are structurally integrated.